So this brings me to the webinar of today. And the webinar of today is going to be given by Paul Wichgers. I'll give a very brief introduction to Paul Wichgers-Schreur. He is a PhD from Utrecht University. He did his PhD on strep suis. Then he joined Wageningen Bioveterinary Research in Lelystad in the Netherlands, where he started to work, fortunately, he switched to viruses, and he started to work on arboviruses, and he pioneered a whole series of new technologies. And it's interesting to say that he specialized and studied in particular the replication of Bunya viruses. And in doing so, he created different variants of Rift Valley fever virus. And he realized that with some of the work he's doing, it can have direct veterinary and medical applications. And that's what he is going to talk about today, especially about a CEPI-funded project called Larissa. It is about a live attenuated Rift Valley fever vaccine for single shot application. It is a huge project that has gone into phase 1 and is now moving into phase 2 in East Africa, and he'll tell us everything about it. It is funded by CEPI with 25 million. He's the coordinator of that project, and I think it's a fantastic example of a One Health project going from, in principle, a veterinary vaccine, into a human vaccine there. But he'll tell you all about it. I discussed with him in advance, so how to do it for the possible questions, and if he can show a lot of slides, and maybe you might on the way you might lose him somewhere. Don't hesitate then to raise your hand. You can do that on the panel. Raise your hand, and then you can actually ask a question in between. But there is ample opportunity to raise questions at the end. Post your questions in the chat if you like, but you can also after the presentation you can also raise your hand, and we can have a lively discussion. So without any further ado, Paul, I would like to give you the floor and listen to your fantastic story. Go ahead, the floor is yours.
Thank you, Albert, for this kind introduction and also hosting me here at this nice webinar series. And as Albert already mentioned, I will talk about Rift Valley fever virus vaccine. I will switch to full slideshow. Do you see the entire slide? Is this okay for you all? No, then I will.
Looks good.
No, it's okay. Yeah.
It's okay?
Yeah.
So, as Albert already mentioned, I will talk about a live attenuated candidate vaccine, which we are developing for animals and humans. We are doing this work, a part of this work, in Lelystad, which is located in the Netherlands, within Europe, and at an institute which contains also a lot of facilities up to veterinary BSL-4, and we are located about 6 meters below sea level. So that's just something nice to know. And we have quite some capabilities to do also challenge infection experiments, which I will show also later, to also in the end test the efficacy of vaccine candidates, not only for this specific vaccine. So, but then directly to the topic, it's a topic Rift Valley fever virus. Rift is an arbovirus. It belongs to the Fenuiviridae family, and recently also within the Bunya virus a group of viruses, there is also a new class. It's a Bunya viridatus class organized now, and this Rift Valley fever virus causes severe disease in livestock, but also in humans. And this I will come back to this later. And it is transmitted by mosquitoes, and a lot of different ruminant species are susceptible for this virus. And at this moment, the virus is mostly endemic in Africa, East Africa region, but also West African regions. And with the temperature rising and mosquito vectors expanding their territory, there's also an increased risk that this virus will emerge to, for instance, Europe or the United States. And then, yeah, this viruses contains segmented single-stranded negative sense RNA genome. It comprises 3 segments, S, M, and L. And these segments all encode for different proteins, of which the S segment encodes for the nucleoprotein, which needs to protect the viral RNA. The L segment for the polymerase to replicate the genome segments. And for this talk, it's most important to remember the M segment, because the M segment encodes for the viral glycoproteins. So the proteins on the outside of the virion, and these 2 proteins are needed for viral entry and fusion with host endosomal membranes. But normally these proteins are encoded by a polyprotein precursor, but we changed the encoding of these 2 proteins. I will come back to this later. As said, this is a mosquito-borne virus, but there are also a lot of mosquito species capable of transmitting this virus. But how does this work in an endemic region? Yeah, this virus really comes with epidemiology of epidemics. So generally, when there is a lot of rain, yeah, more mosquitoes hatch, and some of these mosquitoes are thought to have a virus with them. Could be come from the larval stage, and then after, yeah, the virus is capable of inducing a transmission cycle in livestock or in wild animals, then there is a risk that human can come in contact to infected products or infected tissues of these livestock species. And this may become a huge risk because the circulation of the virus in livestock may be unnoticed to a certain time. And then in livestock generally, yeah, quite some severe diseases can be found, especially in sheep. There is high fever, loss of appetite, general weakness, and sudden death in mainly young animals. This is probably because the virus targets the liver. It can really efficiently replicate in the liver, which then in the end also causes a high level of viremia, which the virus needs to be able to spread by the mosquitoes to new naive animals. In pregnant animals, the virus also targets the maternal fetal interface. And on the right side of this slide, you can see a placentome. This is a ruminant placenta part. And here in brown, you can see the antigen staining in this placentome. So there's a lot of viral antigen in this placentome, which in the end results in abortion. And besides targeting the placenta, also the virus is able to escape to the fetus, and this may also contribute to the abortion storms, which are frequently seen. So this virus is really a virus of young animals, young ruminants, and of pregnant animals. In humans, in general, the majority of infection results in flu-like symptoms. They resolve relatively quickly. However, in a minority of cases, these infections develop into of more severe complications are found, such as retinitis, but also life-threatening complications such as encephalitis and hemorrhagic fever. And there's not a lot known, but there are also indications that maybe also in humans this virus is capable to trigger abortions. However, to really be certain to show this, additional studies have to take place. On the right hand, you can see some human placenta explants which were exposed to the wild-type virus, and you can really see that also there the wild-type virus is capable of infecting human explants. So to control this virus, which was initially discovered in the 1930s, there are already some inactivated vaccines available and also classical live-attenuated vaccines available, but they are only being used in livestock. However, for the inactivated vaccines, generally multiple administrations are needed to induce a proper and sufficient immune response, and they have a relatively high cost of goods. Some very potent live-attenuated vaccines are also in several East or Western African countries available. They are very efficacious, but generally most of these vaccines are not fully safe in pregnant animals. These live-attenuated viruses being used still suffer from remaining virulence. For humans, there are no registered vaccines and there are a few in development. And what I present today is just one example, but there is also a vector vaccine in development, the TiHo platform, and also another live-attenuated vaccine, DDVax, and a messenger RNA vaccine. And in addition to these vaccines, there are currently also no therapeutics available. So there is still an urgent need to develop improved vaccines, and our approach is the rational design of a live-attenuated vaccine. So how did we came to the design of this new live-attenuated vaccine? This was by some fundamental research on the replication kinetics of this virus. And we were particularly interested in how these bunyaviruses, which contain 3 genome segments, how these package their genome, and how these genome segments really end up in variants, and whether with this information we could improve existing vaccines by reverse genetics. And to make a long story relatively short, we succeeded in dissecting this packaging mechanism, and by performing some high-resolution microscopy analysis, we observed that there was no evidence of interactions between these different genome segments, so between the S, M, and L genome segments. So there was no evidence of the formation of a supramolecular genome segment complex, RMP complex. Furthermore, we found that the majority of variants of progeny being produced do not contain these 3 genome segments, but lack 1 or 2 segments. So there's a whole population being formed during the replication of this virus of variants, which the majority lacks 1 or 2 genome segments. However, interestingly, these incomplete variants seem still to work together to be able to deliver a complete genome in vitro and most likely also in vivo. So with this, it seems that the bunyavirus particle population really consists of a huge mixture of a mixture of variants which acts together, and this is really different than the textbook bunyavirus particle and how it was thought these bunyaviruses spread. And all this work has been done by Eric. Most of the work has been done by Eric, and I see he was also attending this meeting. Happy to have you here. So how did we then go from this fundamental knowledge about genome packaging towards a novel vaccine? So we thought that with this random genome packaging, this flexibility in genome packaging, we could maybe encode the glycoproteins GN and GC on 2, normally encoded by the M segment. We thought maybe the virus is capable of also grow when we encode these 2 glycoproteins on separate genome segments. So by using reverse genetics, just rescuing the virus which is encoded on plasmids, we try to to construct this 4-segmented variant. And we succeeded by encoding the GN on a separate segment and the DC protein on a separate segment. Still viable virus was obtained. Furthermore, as the virus also encodes a non-structured protein on the S segments, in addition to this nucleocapsid protein, we deleted this non-structured protein as well, because this is a main virulence factor. And thereby we designed a vaccine which on the one hand has a split M genome and thereby an imbalance in a replication is being created, and also we make it a virus more complex to package the genome. And the second attenuation is then the deletion of this NSS virulence factor, and thereby we increase the innate sensing. So both are major attenuating effects. So of course, for an attenuated vaccine, it's important that it's stable, but also that it can grow to sufficient titers in vitro, so to allow sufficient production. And we were able to grow this virus up to titers of 10 to the 7 in Vero and BHK cells, so in cells lacking an interferon response. As said, we have a double safety, but we also confirmed stability of this virus after 20 cell passages in vitro, and confirmed the phenotype by specific PCRs, but also by next generation sequencing. And we're really able to show that even after 20 passages, the sequences are stable. So with that, we were able to assess also safety in animal models, and before going into humans, we have to do a lot of safety studies. So from left to right, we started off with acute hepatitis model. The wild type virus is really capable of inducing disease within 2 days in these mice. But we observed that the vaccine variant does not cause any disease when applied in this model. A similar model based on the same mice but using a different route. The first was an intraperitoneal route application, but if you would administrate the virus intranasally, generally the wild type virus and also NSS deleted variants go via the olfactory bulb to the brain. And here you can see that the control mice really die within about 10 days, but none of the 4S inoculated animals got sick or died from the infection. Furthermore, additional safety was observed in a nude mice model. So nude mice lack a proper immune system, a proper T-cell response. However, we observed that all mice remained healthy, even with relatively high doses of the virus. And finally, we also performed a GOP toxicity model in rats, using rats, and we observed that all findings were mild, and they were considered non-adverse. But this was all about rodents. As said, the wild type virus is really a virus of ruminants. So we also assess safety in sheep. We performed dissemination, shedding, spreading experiments, and also reversion to virulence experiments, and didn't observe any reversion to virulence, and also no shedding and spreading from inoculated animals towards naive control animals in the same pen. Furthermore, we confirmed safety in the pregnant ewe model. So as said, the wild type virus really targets the maternal fetal interface, but with the vaccine virus, only healthy fetuses were observed. And in marmosets, the blue line represents viremia in wild type virus inoculated animals, which is lasts for several days, and animals inoculated with the 4S variant didn't show any viremia. So also in the marmoset model, high safety was observed. So with all this quite an extensive package of non-clinical safety experiments, we also went to efficacy trials using young animals, young lambs, goat kids, and also calves. And the blue lines all represent mock vaccinated animals. And there we generally observed about 2 days post-challenge, quite a significant induction of temperature increase, a fever is observed, and hyperemia. But in all vaccinated animals, one times vaccinated, no fever was observed and also no viremia. And finally, at necropsy, we only found challenge viral RNA back in the mock vaccinated animals. So this vaccine is not only safe, it also induces a very potent immune response and protective immunity in ruminants. And finally, we also assessed safety of efficacy of this vaccine in the pregnant ewe model. And there we observed that mock vaccinated animals all aborted around 1 week post-challenge. And you can see a lot of bloody placentas. These placentomes are disintegrating, and we observed a lot of viral antigen in the livers of the fetuses. In the vaccinated group, no abortion and all live fetuses were observed, and also no evidence of viral replication or viral antigen in the organs of the fetuses. So that all this preclinical package also started more or less the work for testing this vaccine in humans. And this was really facilitated by a CEPI grant already back in 2019, in which we started pre-COVID. And there, with a huge consortium as depicted here, we started to prepare for a phase 1 clinical trial. And a major part of this Larissa 1, I referred to it, was manufacturing of this vaccine under GMP conditions. And at IDT Biologica, a German CRO, we succeeded in the manufacturing of the first GMP batch of this virus. And this was GMP lot was produced by Virocells in cell stacks, and using some standard clarification steps, but all steps had to be optimized. And with this GMP lot, we were able, in collaboration with CIVAC, we were able to assess safety, tolerability, and immunogenicity in healthy adults. And we started. The CIVAC is located in Belgium, so we started with the naive individuals, of course. And in this first in human clinical trial, we tested 4 doses in 3 different cohorts. So we started off with a dose of 10 to the 4th, followed by a middle dose, 10 to the 5th, TCID50, and ended with a high dose is 10/6 TCID50. And in between these cohorts, there was extensive safety monitoring and review whether we could go to the next cohort. And quite some sampling, we did quite some sampling also to ensure that we really monitor also potential dissemination of the vaccine virus beyond a certain vaccinated individual. So here are the main outcomes of this first in human clinical trial considering safety. And we observed that the vaccine was really well tolerated, and solicited adverse events were generally mild and transient. So within 23 days, 72 hours post administration, most transient of most adverse events resolved. And these mainly consisted of pain at the injection site of a relatively low grade, but also we observed we observed fatigue and headache in half of the individuals, but this was also very short-lived, and this is something which is in line with a lot of other vaccines, and no, it didn't worry anything. Moreover, we collected blood, urine, saliva, and semen samples and assessed these samples for viral RNA and didn't found any vaccine viral RNA back in these samples. So really showing that this vaccine virus does not disseminate efficiently beyond an individual being vaccinated. But it's not only about safety, we also assessed the immune response. So started off with some humoral immune responses. So on the left, you can see the graph in which we present nuclear capsid antibodies. And this is being performed by a semi-quantitative ELISA. And as you can see that in time, especially at 2 and 4 weeks post vaccination, not a lot of differences between the dose groups are being observed. All individuals seem to respond well. But later on, after a half year and a year, there are a bit more differences between the dose groups, and the high dose group, the green line in this graph, is really outperforming the rest. Quite a different pattern is being observed with in the VAT, in the virus neutralization test. We observed much more of a dose response. So early on after vaccination, at 2 and 4 weeks post-vaccination, especially the high dose group really outperformed the middle and the low dose. And all individuals in the high dose group responded, and about half in the middle dose and only a few in the low dose. What we also observed was a decline in these systemic antibody levels, and we are still, we need to investigate what this would mean from a long-term protection perspective. And at our partner at TiHo, App and collaborators, they assessed, in addition to the humoral immune response, also T-cell responses. So here are ELISpot data being presented, and PBMCs were stimulated with peptide pools of different viral antigens. So on the left side, PBMCs were stimulated with peptides from the nuclear capsid protein N, and on the right side of the glycoproteins. And here we also observed that that was not quite a lot of not a significant dose response. There seems to be a similar response over dose group for the nuclear capsid protein. However, a more obvious dose response was observed by stimulating with the GN-GC peptide pools, in which the high dose group seemed to outperform the rest. And again, later on, after half a year, these responses declined, or at least we could not measure them anymore. But we are still we do not know what this would mean from a protection perspective. I will come back to this later. And some very exploratory work was also being done to assess whether these T-cell responses are CD4 plus or CD8 plus related. Now, we observed that in several individuals, in the majority of individuals, this is the interferon gamma responses were observed in CD plus and CD8 positive T cells. And also some work is ongoing to really assess the T cell memory, and so far we get some promising results there. In addition to humoral and T-cell responses, we're also interested in early innate immune signatures. So during this first human clinical trial, we also obtained some PAX-gene RNA tubes, in which we were able to assess host transcriptome the host transfer control, and we used a nanostring assay, maybe familiar for some of you. And we really were interested in what kind of innate immunity genes will go up or down following vaccination compared to the placebo. And very fresh, or more or less fresh results, we observed a strong induction of interferon responses in all individuals that received the vaccine. And we had a perfect grouping, as can be seen on the right, for the placebo and vaccine at 1 day post-vaccination. But also good to mention is that this interferon response was short-lived and resolved more or less resolved by day 3. And recognition of a live attenuated vaccine by the innate immune system is a good thing on the one hand, because then also you would expect activation of the adaptive immune response. And this is also what we saw by maturation markers being present at day 7. And furthermore, I can now mention that we did not saw any signs of gene signatures which would be associated with vaccine safety concerns. So most of the work I just presented is now also being published in the Lancet Infectious Diseases, and the overall message is that the vaccine was well tolerated. As said, no vaccine viral RNA detected in blood, urine, saliva, and semen samples, and the induction of a promising immune response. And with this, phase 1 data coming in, we also succeeded in strengthening our collaboration with CEPI. And we opted for a new project, and we were successful. So currently we are running Larissa 2. And in the Larissa 2 project, we have still a lot of consortium partners, quite some consortium partners are involved, and we really focus on optimizing and scaling the manufacturing. So we hope now to go from these cell stacks to bioreactor process, and we're also planning for the production of a new GMP lot. And as we are moving on in development, it's becoming even more important to increase our understanding of the correlates of protection. For Rif Valley fever virus, it's compared to other viruses a bit understudied, and still a lot to be learned there. And in the end, it's good to mention that we should also already prepare for efficacy trial, which we will do in the epidemiological modeling work packages. But most important of Larissa 2 is that we aim for a phase 2A clinical trial in the endemic region in East Africa. But before we can start, we already performed some passive transfer experiments in which we we purified IgGs of the first in human clinical trial, and we administered these human IgGs in mouse, which were challenged, but also in marmosets. And we aim to define some surrogate of protection. So whether we could more or less determine a VNT titer, which could be considered protective. And doing so this is our really preliminary results, we observed a very strong dose-dependent reduction of virus dissemination. So groups of mice were administered with different IgT amounts, different VNT positive of different neutralizing titers, and observed that especially at a titer of 25, a lot of dissemination of challenge virus was prevented. So almost a 3-log difference between the placebo IgGs. And later on in time, this was a day 2 post challenge. Later on in time, we also see major difference in body weights, whereas animals which received the titer of 20 or 12 were more or less fully protected, and animals receiving a lower titer also developed disease. And this was also reflected in the mortality curves as presented on the right. A similar overall pattern was observed in the marmoset. So we used the same pool, but then administrated to marmoset as a most as a non-human primate, which mostly reflects human disease. And we saw a strong dose-dependent reduction of fever, liver disease, and viremia, especially compared to, if you would compare the placebo group, where you can see quite some temperature increase following challenge, compared to the high dose group. In this case, there was only no temperature increase, and also about even more than 3 log reduction in viral titer. And we're currently performing some virus isolation experiments to also show a strong reduction in infectious virus in this model. But still on the planning. This is just to highlight that we will also perform some non-clinical work with a more relevant challenge. So we aim I think next year or the year after to also assess vaccine efficacy in a mosquito challenge model. And we in the past already optimized some models to challenge sheep by infected mosquitoes. And we were capable of inducing disease highly similar to needle-based disease. So I'm really curious about this model and whether the vaccine is capable to protect there as well. And then some few words about the clinical trial, which we aim to start in 1.5 years from now in Uganda and Kenya. So in collaboration with Veterinary and the UVRI, we aim to test this vaccine in a phase 2A clinical trial, and hopefully also in this endemic region, this vaccine will be considered safe and efficacious. But to already prepare for efficacy trials, which are quite challenging for vector-borne disease, we are really focusing together with a lot of other partners to think about how to measure vaccine efficacy in the end. And there, the surrogate of protection experiments, which I presented a couple of minutes ago, are really helpful as well. And because this virus infects ruminants, animals, and humans, it's very, very important to keep track of all of this and assess the role of the animal reservoir towards the exposure to humans. And as said, these mosquito vectors are expanding territory. It's really important to also pinpoint to what climate and vector distribution, to what extent these play a role. So with that, I'm already coming to the main take-home message of this webinar. So I hope I could show you and convince you that this vaccine virus shows promising results in all non-clinical studies performed. So we observed single-dose protective immunity without safety concerns. And with the first in human clinical trial, we observed that this vaccine is well tolerated, induces B and T cell response following a single vaccination, and that the longevity of the responses should be further investigated. And as Albert already said at the beginning, Rift Valley fever virus infects animals and humans, and with this vaccine we think that this vaccine is really suitable for a One Health approach, so protecting animals but also protecting humans. And so with that, I would like to thank a lot of people. There are too much people involved, so I listed all the partners only. And many thanks to CEPI, who funded this work, and to CIFAC, who performed the phase 1 clinical trial. And with that, I would like to give the word back to Albert.
Well, thank you, Paul, for a fascinating story, going all the way from your fundamental interest in Bunya viruses and really going the translational way and all the way today up to a phase 2 trial. So this presentation is open for discussion, and you can raise your hand or post your questions in the chat. Perhaps, Paul, if I can start.
Yes.
Well, basically, you started to say that CEPI is funding this, and there's basically 3 other vaccine candidates. Yeah, so it's the messenger RNA and the ChAdOx, and also the other live attenuated vaccines. So how do you position this particular vaccine versus the other ones?
Yeah, good question. I think the ChAdOx vaccine, this is a adenovirus vector vaccine. This it's headed by Professor George Varimmer. They are already advancing towards phase 2 clinical trials. So they are a bit ahead from their clinical work, but it's quite a different vaccine. And as you know, also there are quite some challenges with ChAdOx vaccine, considering potential pre-existing immunity and maybe also some safety aspects. So in that sense, to having several vaccines available would be a good point. Considering our differences with the DDVax, this is another live attenuated vaccine. This vaccine lacks another virulence factor, which is the NSM protein. I haven't talked about this considering time. This is a key factor in which reduces potential vaccine replication in the mosquito. But yeah, it's to be seen which live attenuated vaccine would have the best safety profile and the best efficacy profile. But to my knowledge, the DDVax vaccine still has to go into humans. So their first in human clinical trial has to be performed. And the messenger RNA vaccine is still in early development. But yeah, not sure what the future brings. Could be that, like in other fields, the messenger RNA vaccine will go faster. However, I think it would be right complex to express all antigens on a messenger RNA vaccine because this GN and GC protein are quite large, and to encode this into a single messenger RNA is not an easy task. It's not like the spike protein of SARS.
Okay. Well, there's some questions coming in the Q&A, and the chat apparently doesn't work so well. So use the Q&A. There's one question here. The first one here is anyone testing adjuvanted Rift Valley fever glycoproteins as vaccines antigens? Is it possible to make a subunit vaccine with a proper adjuvant? Probably.
Yeah, that's. I've also been involved in another more model-orientated project called SAPI. What's IMI funded, and there also the subunit approach was being assessed for this virus. I think it's very well feasible, but for all subunits, you would need a double vaccination regimen. You need to have two vaccination with a relatively short interval in between, and maybe also a booster vaccination. So it's I think it's
that might apply to this vaccine as well. Yeah, you're not completely sure that the memory response will be sufficient, and you might have to give a boost.
Yes, but I think we are aiming to give this boost later on after several months. So not after 3 weeks. And for a response during an epidemic, you would like to see some protection with a single shot already, even if it's short term, for a couple of weeks, months. And for a subunit, at least what we saw with purified GN glycoprotein administered with an adjuvant, then it's not you will not get sufficient neutralizing antibody titers, but may depend on the type of adjuvant and the dose being applied.
Perhaps especially since CEPI really wants to have the vaccine ready as soon as you have an outbreak, for outbreak management. That's my my perception of the whole story. And for that, it's different, like for SARS-CoV-2, for instance, where you have a circulating virus, whereas this gives a concentrated epidemics. Is that correct?
I missed your question up. Sorry.
Okay, sorry. So basically, what you want to do is in an outbreak situation, yeah, because that's what that's what SEPI wants, a vaccine for rapid response, yeah, and with a live vaccine, you might have a better chance there, yeah, so that you interfere faster than with a subunit approach. I think that for this particular vaccine, it may be true.
And maybe also cost of goods might come into place.
So there's some more questions there. I still have a burning question for you as a fundamental virologist, Bunya virologist. If I look at the situation with the segmented genome viruses, the influenza virus, we're always afraid of reassortment. What is there any risk of using this in the field, that you get more dangerous reassortants?
I think the risk is very, 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 very low, although not zero. And this is argumented by that we have modified the M segments, so into 2 M segments which lack a gene which is needed for spreading. So if wild type virus would gain such a half a M segment, it will not gain anything. For the S segment, it's the same. It lacks the NSS gene, which is a virulence factor. There's no gain of a wild type virus to get this segment and to spread more efficient. For the L segment, yeah, theoretically that would be possible. However, yeah, this is highly unlikely because this vaccine virus does not induce viremia, as we showed. The chances that in a single cell a vaccine virus and the wild type virus come together is neglectable low, I would say, but not zero. So
yeah, and at the end of the day, you cannot expect this to be a more serious variant than the wild type. Is that correct?
Yeah, with the information about Rif, you cannot expect this. No, indeed.
Okay, I have a question here from Etrewiki. So usually animal viruses are not sufficient titer to allow cooperation of variants with complementary genome segments, as happens with plant viruses. Yeah, is the assumption that high titers of virus are injected by mosquito that allows multiple particles to infect one cell? I think it's quite an interesting one when you talk about these incomplete viruses. And now you're actually coming in with an overcomplete virus with 4 segments.
Yes, that's from a vaccine perspective. But if you focus the question on the wild type virus, I would say it's highly likely, although we have no experimental evidence, is that also in a mosquito these variants act together. So in saliva, maybe different variants containing incomplete genomes act together and co-infect a single cell and thereby deliver the entire genome of this virus. And there are some really nice additional papers coming from the multipartite plants work that also show that some plant infecting Bunya viruses seem to use this strategy as well. So it's really complementing what we are seeing, and this is quite a difference in the field of in the perception of how these viruses spread. It seems that these segmented Bunya viruses really differ in how they act as a virus population.
Because theoretically, this could be an issue with influenza viruses as well, but you don't see it.
I just this afternoon I read some additional papers and saw that also for influenza virus there is evidence that not all viruses contain all 7 of all 8 genome segments, and sometimes also no expression of some viral genes is not being evidenced, and therefore also yeah, the suggestion is that sometimes also co-infection takes place in which just the missing segment will then be complemented. It's not as obvious for influenza. You need of course some kind of selection, because otherwise the virus would end up with too many particles which are incapable of spreading. But for a 3-segmented virus, this is something in between.
Great. So one issue you did not address is the antigenic variation of this particular Bunya virus. Is it, let's say, if you look at influenza, if you look at many of the other segmented viruses, there's a lot of variation. So can you do with one vaccine? Yeah?
Yes, I think so. It's because there's only one serotype being described for Rift, and the difference between genotypes is relatively low. It's highly conserved, and which I also maybe didn't talk about briefly, but in many of these non-clinical animal experiments, we already challenge with another genotype. So they all have the same serotype, and we challenged with another genotype, and also did some cross neutralization assays, and there we do not see any differences between strains. In other Bunya viruses, there are more differences within the species. So it might be just the luck from a vaccine developer perspective that there's only one serotype.
Well, that at least that is some good news there. That indeed you can go with probably with one vaccine without any further escape. There's another question here. It would be it would be interesting to see if mosquitoes can transmit if the mosquitoes can transmit the vaccine. That's another possibility.
We are currently doing these experiments, and I can already with initial pilot experiments already being performed, and we can see a great reduction in the capability of this vaccine virus to be replicated by the mosquitoes. And we are assessing needle inoculation and also blood feeding inoculation. But this is, we will do this work the coming months. It's very good point. It will also add to the safety profile if we would see a reduction.
But if you don't get any viremia in the vaccinees, is it a relevant question?
Okay, yeah. I think it will only add up to the safety profile. If you would be able to show or state that also the vaccine virus, in the very, very, very rare occasion that it would be taken up, end up in a mosquito, that it is unable to disseminate in the mosquito, this would make the story even stronger.
Okay. There is a question here by Paul Hodgson from Vido. And he says, sorry, I may have missed it, but the question is basically about the longevity of protection there. Yeah, so does the disappearance of the antibody, does that mean that your protection is gone? And it also relates to correlates of protection, of course, and memory cells, because it doesn't mean if you lose your neutralizing antibody titers, and taking for granted that neutralizing antibodies as the main correlate of protection. Yeah, I'm a little bit uneasy there, because within a year you don't have you don't have the neutralizing antibodies anymore.
Yeah, we do not have the proven protection, because we know neutralizing antibodies correlate with protection. Unfortunately, we do not have duration of immunity data for half a year from pre-clinical work. That's something we eagerly want to do. However, it's those are expensive trials to do, because you have to keep these animals quite some time. But it's a very valid point, and this is definitely which I'm I try to arrange and get funding for.
Perhaps a last question there. Basically, that's a One Health question, actually. Yeah.
Oh yeah, sorry.
So why don't you vaccinate the animals if you eliminate the virus from the animals? So there's no more problem. And this is a more generic thing, of course. We can talk We can say the same thing for H5N1 influenza. So what is your idea there where you have a vaccine that can be used both in humans and animals?
If you would succeed in really effectively administering vaccines to the ruminant population in these endemic regions, then okay. If you have a coverage of 80%, 90%, then this would definitely be a very suitable approach.
Okay.
However, I think this is not always realistic, especially in those settings, and the incentive to vaccinate is also a bit difficult because the vaccine of the wild type virus, the problem is not always there. So you also need some incentive for vaccinating livestock. Therefore, in the veterinary field, there's also some discussion about using combinational vaccines. So not only Rif, but also PRRV or other viral diseases which you can combine in a single vial. But still, this is difficult. And that being said, then it's possibly better to be able to protect people at risk with a human vaccine, so slaughterhouse, of people working in slaughterhouse or cattle rearers, and in addition have a vaccine available during larger outbreaks.
Yeah, it is actually what you said is quite important. I think that it really depends on the setting as well. I may remind you of the fact that we eliminated rabies in that way from Europe at least, or at least Western Europe. Yeah, so there is an issue there, but rabies was a relatively easy one there. So I think indeed the complexity of the regions you're talking about make it more difficult. But the good thing is you have a vaccine for both. I'm afraid we are running over time.
Okay.
I think fantastic presentation and a fantastic example of how One Health approaches, from fundamental research into animals, into humans, eventually how that indeed results in new vaccines for the future to combat these diseases. So Paul, thank you very much, and I would also like to thank ISV and the whole audience for staying on board for so long. And it was a great presentation. Thank you.
Thank you all.